Hello everyone, welcome to experimental biochemistry course. In this week we are going to study protein ligand interaction. Now from the start of this course we have seen different characteristic of protein. In the theory classes we have studied how protein structure has been made, rather how the primary structure of the protein folds to form a 3-dimensional 3D protein structure. Secondly, how proteins are expressed, purified and characterized. This have been studied both in the theory as well as experimental courses, and most importantly we have seen the enzymatic activity of protein. Now the enzymatic activity of the protein is very much important, as we all know, because it determines whether the protein will function or not in a biological medium. However, when we are talking about a biological medium, what we can see is that there are various other solutes in a biological medium apart from the ligand of our choice, or apart from the substrate, or apart from that compound which protein will catalyze. Briefly, what we can say, in a cellular environment, as we have seen, or rather schematically seen in various textbook, in a cellular environment various sorts of cell organelles are present. Apart from the cell organelles in the cytoplasm, various solutes are present, starting from salt to sucrose molecule to lipids, and all other things are present within a human biological system. So when protein is present within a biological medium or within a cell, so it does not interact with only one particle at a particular time. What we can say in a more easier term is that when you are considering a particular interaction with protein and a particular ligand, okay, so protein and ligand coming close to one another, through ligand what we are trying to express is that any sort of solute, any sort of molecule which will interact with protein. So in a biological medium, when a protein and a ligand or any molecule come close to one another, it is prone to some sort of interactions. Coming to those interactions, those may be, some may be specific and some may be non-specific. Regarding specific and non-specific, those are a bit advanced, which we will not discuss right now. However, what we can say is that from your previous classes where you have studied about the enzyme kinetics, those interactions might be somewhat specific. Why? Because the enzyme interactions happen in such a way where the molecule or the substrate attaches to the catalytic site of the enzyme, and in the catalytic site there are specific residues which basically react with the substrate and catalyze it to form a specific product. Now suppose a particular molecule or particular substrate, in spite of attaching to that active site of the protein, attaches to somewhere away from the active site. Well, it might happen. The region which is which contains the active site of the protein might not interact with that particular molecule. The molecule might attach somewhere away from the active site. Okay. In that cases, the interaction is not that much specific. Well, we can't actually define this as non-specific right now, because it will be much more generalized. On a easier note, what we can say is that those interaction which does not attach in the active site, where the molecule does not attach in the active site, they attach in somewhere away from the active site. Fine. So where it attaches from away from the active site, we might be interested, and this gives rise to a new kind of study known as a protein ligand interaction, where a ligand attaches to a region of a protein which may or may not be the active site, or which may be far away from the active site, and we are interested to find how the interactions can be determined. In order to determine such interactions, various studies have been done. Some may be spectrophotometric methods, some may be other methods. Some studies you have seen in the theory classes. In this experimental class, what we are going to see is that how we can promote UV-Vis techniques in order to basically find the mode of interactions, or basically quantify the interaction. In order to quantify the interactions, we need the particular protein, a ligand, and then we will spectrophotometrically determine how the interactions can be mathematically evaluated. In order to study the protein ligand interaction, the protein of our choice is a very well known serum albumin protein. Now there are two different types of serum albumin which is commonly studied in biophysical chemistry classes. One is human serum albumin, or commonly known as HSA, and the other is bovine serum albumin, commonly known as BSA. Now, the serum albumins are quite familiar with us because we have used it in our previous classes, where we have studied the spectroscopic determination of proteins, amino acids, and also in protein denaturation and folding studies. Now, we are going to use human serum albumin. Why we are interested in serum albumin all of a sudden? Now, serum albumin has a practical role in our biological system. In terms of serum, we might be quite familiar that it is present within blood. Okay. So, it has various function, and one of the important function is that it maintains an osmotic pressure within our body. That is a bit detailed mechanism which we might not cover out here in details. On a gentle note, what we can say is that when we are talking about HSA, human serum albumin, it is basically one of the transport protein which we can consider. Regarding transport, it is a bit generalized term. So, when something goes in our blood, any sort of medicine, or when any injection is pushed or any medicine has been injected within our body or within our blood. Okay. So, what happens is that those molecules or those medicines bind to some proteins. Okay. So, those proteins basically carry those molecules to the target site. So, out here what human serum albumin can do is that human serum albumin can bind with those medicines, those proteins. It can bind in the blood system, and it can carry those molecules to the target site. Fine. Now, suppose we take some molecules which do not bind with the human serum albumin, and we inject those molecules. So, it will be completely irrelevant because it will not bind with any protein, and it will only get disintegrated in the bloodline. So, how to determine whether a protein or will interact with a particular molecule or not? For that, we first need to see whether that particular molecule or that particular ligand which should be injected or which is biologically important, whether interacts with the protein of interest, that is human serum albumin. Now, the two compounds or the two ligands which we are interested in studying is that one is gallic acid out here, it is seen as gallic acid, and the second one is curcumin. Fine. So, curcumin, many of you might be familiar with curcumin. It is present in turmeric, okay, that is we regularly use in our house. Fine. So, that yellow powder is actually curcumin. Curcumin has a lot and lots of application. Okay. Starting from our health benefits, it has antioxidant activity and it protects neurons, and all sorts of good benefits are present in curcumin. Similarly, with gallic acid, it is also beneficial for our body. Now, when you are taking curcumin or when you are taking gallic acid not in pure form, okay, so we are generally not take this in pure form because of certain solubility issues which we will address a few minutes later only. Okay. So, talking about curcumin and gallic acid, when they are injected or when taken in the body or in the biological system, so whether it is interacting with the serum albumin or not, we are interested in. For that, we have chosen these two ligands, and we will taking a protein, that is human serum albumin, and the medium which we will use is phosphate buffer pH 7.4, and the concentration is 20 millimolar. As all of you might be familiar that phosphate buffer basically used to mimic a cellular environment. So, we are using phosphate buffer out here. Now, these two, what we are going to do is that we are going to estimate separately. First, we are going to estimate either gallic acid, and then we are going to compare this one with curcumin and see which one interacts in a better way with human serum albumin. Now, let us see the amount of gallic acid and curcumin we require in order to carry out our experiment. Now, this is gallic acid. We are going to prepare 5 millimolar 25 ml gallic acid. I have calculated it, and the weight is around, as per your calculation for molarity, the weight is around 0.021 gram for 5 millimolar 25 ml gallic acid. Now, here it is the weighing balance. Okay. So, we have kept a butter paper out here and we have tiered it. So, the value is showing 0. And now, what we are going to do? We are going to add 0.021 gram of gallic acid. We are going to add a pinch of this gallic acid now. Okay. So, when we have added a small pinch of this gallic acid, what we can say is that the weight has reached to almost around 0.0253. So, it is a bit higher than 0.021 gram. However, we can easily proceed with this one, because as we have discussed previously, we should take the amount a bit higher than the required one. Now, we are going to transfer this amount of gallic acid to a falcon tube. Here I have taken a clean falcon tube, okay, it is a 50 ml falcon. Now, this butter paper I have taken gallic acid out here, and I am going to gently put the gallic acid here, okay, a small amount of gallic acid out here, okay, as we can see this white powdery substance. To it I will be adding 25 ml double distilled water. Okay, you might be dissolving this in various solvents, but for biological experiment kindly dissolve it in double distilled water. Okay. So, I am adding double distilled water. Initially, after adding 10 to 15 ml of double distilled water, I will just put this cap and will gently mix it. So that it dissolves completely. It might not dissolve completely out here. A part of it has already dissolved, however, a small portion is still there, a small portion of the solid. Okay. Now, I will add the remaining amount of with double distilled water. Now, I am adding here 25 ml double distilled water. Okay. Now, after adding 25 double distilled water, I will again just stir it a bit, mix it thoroughly, and now as you can see, a small amount of gallic acid is still undissolved out here. Okay. So, what does this show? This shows that gallic acid is not completely soluble in water at this state. Okay. So, before taking the concentration of gallic acid, kindly note down its solubility. However, right now what I will do is that I will give it for sonication in ultrasonic bath, and after some time it will completely dissolve. Okay. Now, advanced experiments have been made, various sorts of experiments have been made from time to time in order to solubilize this gallic acid. Due to its poor solubility in this aqueous medium, sometimes taking of this gallic acid into the biological system is not at all beneficial, because it is not soluble within the aqueous medium. So, it should be made soluble, fine. So, that is a bit advanced research, but however, for the time being what we can say is that it is not completely soluble in water, gallic acid. So, we have to give it for sonication so that it mixes thoroughly. Okay. So, I will giving it for sonication and we will proceed in measurement of curcumin solution. We have measured gallic acid and made gallic acid solution with using double distilled water. Now, we will measure curcumin. We will prepare around 20 millimolar 10 ml curcumin solution. Why 20 millimolar 10 ml? I will discuss a bit later only. For that, we require basically 0.073 gram of curcumin. Okay. So, here is curcumin. Now, I have teared it, we have kept this paper out here, and the way we require is 0.073 gram. So, we will take a pinch of this yellowish powder . So, it is around 0.03 gram. I require just almost double this weight. Okay. Now, it is 0.0751 gram. Okay. So, let us take this out for convenience. Now, this one is the yellow powder or curcumin powder, what we can see. It has a very distinct color. Okay. So, it is very interesting to see because the turmeric we used in our home has the same color. Okay. If you paste turmeric, it has the same color, fine. So, curcumin, it is actually the curcumin which we are using, it is this color. Now, curcumin are of various grades when you purchase it commercially. Curcumin has various mixtures, also mixture of other compounds. Okay. So, it is better to go for some high graded curcumin compounds. Okay. Now, this curcumin, what I will do is that I will dissolve it not in double distilled water. Okay. Remember, not in double distilled water, but in ethanol, fine. So, I will dissolve it in ethanol. So, we have dissolved the curcumin in this falcon tube in ethanol. Now, there are various grades of ethanol which we generally use. Okay. The grade which we use out here is spectroscopic grade, or rather HPLC grade, as you can see here. Always use this type of ethanol while making any particular chemical solution. In laboratory, you can see various other sorts of ethanol which do not have written this analytical grade or spectroscopic grade. Do not go for that ethanol, because that ethanol is used for other purposes like washing or sterilizing, all sorts of things. However, out here the ethanol which you will be dissolving it is in this type of ethanol. So, this is the solution which we have used. Okay. So, here you can see it is an intense yellow color, fine. Now, we will not use this solution for our analysis. What we will do is that we will take a small amount of this solution, around 5 ml of this solution, in a falcon tube and make it the volume to around 25 ml, fine. So, why we are doing this? Basically, curcumin is not at all soluble in double distilled water. You can try it by yourself. You can add water to curcumin powder and you can see that it is not solubilizing in water, fine. Whereas, on the other hand, it is soluble in ethanol to a certain fraction, but of course, this one is much higher than that of double distilled water. So, what we do, we basically take higher concentration of ethanol and higher concentration of curcumin out here, fine. Higher concentration of ethanol means it is pure ethanol without any dilution, and higher concentration of curcumin means we are using 20 millimolar of curcumin. For that, we are going to prepare in actual 5 millimolar of curcumin from here. For that, we are going to take a small amount of this ethanol, curcumin dissolved in ethanol, and we are going to transfer it in a falcon tube containing double distilled water. Now, what is happening in this process? So, the final ethanol solution has basically, the final curcumin solution has basically ethanol water mixture, fine. So, the solution which we are using for analyzing this protein ligand interaction has water ethanol mixture or ethanol water mixture. However, please keep it in mind, the solution which you will be using for protein ligand interaction should not be pure ethanol. Why? Because you have studied previously in the theory classes that ethanol basically degrades the protein, it denatures the protein. The protein structure will disintegrate when you will increase the concentration of ethanol from time to time in a titration method, as we will see in a short time, fine. So, there is again a limitation of solubility in this. For that, what we are doing is that we are taking a higher amount of curcumin, dissolving it in ethanol. Now, we will take a small amount of this ethanol dissolved curcumin, okay, and will transfer it into another falcon. Okay. Now, we will prepare 25 ml of curcumin solution in ethanol water mixture. For that, what we are going to do, we are going to take out 5 ml of ethanol into this, and the total volume will be 25 ml. Okay. So, 5 ml of this ethanol curcumin mixture in this falcon and making up this volume with double distilled water. Now, this is a 1 ml pipette. I have adjusted the volume up to 1 ml, and now I will gradually take this curcumin solution from here and will transfer it into this falcon. Okay. 1 ml, 2 ml, 3 ml, 4 ml and then 5 ml. Okay. Now, to it I will be adding double distilled water. Okay. Now, it is almost around 25 ml solution. Yep. Now, this curcumin is actually 5 millimolar curcumin dissolved in double distilled water, or rather in ethanol water mixture. Please note, the percentage of ethanol in your solution should not exceed around 20 to 30% of ethanol percentage. Okay. What happens actually if ethanol percentage increases in your solution? In that case, the protein again will start malfunctioning and it may degrade. So, better keep the ethanol percentage low. Out here we have kept around 20 or less than, a bit less than that. Okay. So, we are proceeding with this one. This is curcumin dissolved in water ethanol mixture. Now, we are going for measurements. For measurement, what we will do, we will initially proceed with UV visible spectroscopy. Okay. For UV visible spectroscopy, we will going into details why we are going into UV visible spectroscopy, but before that let me just give you a brief recap about the UV visible spectroscopy. Now, this UV visible spectroscopy has uses two cuvette, as we all know. So, here this is the transparent side of this cuvette, and this one is actually a 3 ml cuvette. Okay. So, we will be taking phosphate buffer solution and the volume will be 3 ml out here. So, initially we are adding 1 ml. I am taking phosphate buffer and I am adding here 2 ml, and number 3 ml. Okay. So, as you all know, in a UV machine, as it is shown here, these are quite familiar because we have studied it and we have used it for our previous experiments also. This UV machine has a chamber out here, it contains 2 compartments. Okay. So, now I will take this one and keep it in one of the compartments. Now, before keeping it in a compartment, we should wash both of these transparent sides and keep it in such a way that the incident light faces the transparent side. Okay. Similarly, I will take another cuvette. Okay. The first one I have kept it in the blank chamber, and now I will keep it in the sample chamber. Now, I am washing this one, okay, this 2 sides, 2 transparent sides I am washing, and I am holding the cuvette in the other side. Okay. Do not hold the cuvette in the transparent side. Now, I am placing this one again in the sample part, sample holder. I am closing this lid, and out here this is the software. We are all quite familiar with this software out here. So, in the software what we can see is that this is the method part, here method. Okay. We can go here, we can see that it ranges from 200 to 600. This is the wavelength range over which the scan has to be carried out, fine, and the scanning speed is kept as medium. Okay. And the scanning interval is 1 nanometer, that is, it will take reading at each and every single nanometer. Okay. And the file name I have selected a file name. Okay. And the rest of the parameters should be kept same, and now I am clicking OK. After this one, when we have kept the phosphate buffer in the 2 cuvettes within this UV machine, first we will go for baseline correction. So, we will click here this baseline. Once we click this baseline, it will show the start wavelength as we have given here 600, and where it will finish it is 200 nanometer, and we will click OK. So, baseline is actually getting initialized. Now, for the time being, why we are interested in analyzing this UV spectroscopy. Okay. So, what happens is basically we are going to take the molecule either curcumin or gallic acid in the cuvette, okay, and we will measure its UV spectra. Gallic acid or curcumin has a separate UV spectra which you can see in any sorts of paper, any textbook or online. Okay. So, we will do is that we will add to it HSA or the protein of our interest dropwise in a titration method. What does that mean? That actually means is that we will be taking curcumin in a cuvette. Okay. We will take its UV fine, it will give a characteristic UV spectra of curcumin or gallic acid. To it we will first add small amounts, say 5 microliter or 2 microliter of HSA solution. The HSA solution which we have prepared actually, we will add it, and then we will see how the spectra changes. Okay. Which spectra changes? We will be interested in the spectra for gallic acid or curcumin. Now, why we are interested in this case? Because what happens, gallic acid or curcumin has a specific spectra, UV spectra, and now we know at this stage that UV spectra or absorbance spectroscopy gives an insight into the chemical environment of the compound also. Now, we know basically from our spectroscopic studies or UV spectroscopy, which many of you might be familiar with, that when we are changing a molecule from polar to non-polar environment, it shifts, basically the lambda max shifts in either in the red or the blue region depending upon the medium, fine. So, what happens is basically when the environment or the chemical environment or the surrounding of the molecule changes, this UV spectra is prone to particular change. Now, in this case what you can say is that it is kept in a environment, first is phosphate buffer, okay. It gives a particular spectra, and now when protein is added to it, its medium changes. Now, why this medium changes? Because this curcumin might attach to the HSA or it might not attach properly. However, its medium is prone to change because we are adding a foreign substance, that is protein, to the phosphate buffer medium. So, its UV spectra might change, and again when you are gradually increasing, we are getting a change in the UV spectra. Depending upon the intensity of these changes or the magnitude of these changes, we can basically quantify the amount of interaction between the protein and a ligand, and that is why UV spectroscopy serves as an important tool for determining protein ligand interaction. I have taken out this cuvette which was kept in the sample holder, fine. So, we will take curcumin solution into it. First, what I will do, I will take this micropipette. I have adjusted it to 10 microliter. These are 20 microliter micropipette. Okay. So, I will be initially taking out 20 microliter of this solution from here, okay, into a waste beaker, fine. So, what I am doing is that I am basically preparing this curcumin solution in a 3 ml phosphate buffer solution. When we will be adding directly 10 ml curcumin to it, then the total volume will be 3 ml plus 10 microliter. In order to avoid that, what we are doing is that we are initially taking out 10 microliter first. The volume is actually 3 ml minus 10 microliter. Then I am again adding curcumin, that is 10 microliter curcumin, to make the final volume to 3 ml which was in the first. Okay. So, this one is basically curcumin solution in water ethanol mixture. First stir it gently, okay, or rather mix it thoroughly before using it. Then I am going to take 10 microliter of this curcumin. Okay. I am going to take 10 microliter of curcumin and add it in this phosphate buffer solution, and I am adding this one in the phosphate buffer solution and mixing it thoroughly. As I have previously discussed in the spectroscopic related classes while taking this experiment, while doing this experiment, that please note air bubble or rather water bubble is not formed within this solution or within this cuvette. Okay. So, once we have taken it, take this cuvette and put it in the sample compartment. Okay. After that, we will go for observation. So, I have taken this curcumin within the cuvette, okay, and now I will do auto zero, but before that what we can see a small amount of absorbance value is being shown. In order to remove that, we will do auto zero. This auto zero basically fixes the initial wavelength to 0. So, it is around 0.01, okay, we can proceed with this. So, it is now 0. So, we have clicked start, and let us change this one to around 0.1. So, here we can see absorbance taking place, the increase in the absorbance value out here. The value again decreases a bit. And out here this fluctuation is because of the change in the lamp. Now, it again increases. What happens is basically around 340 or 350, the lamp changes from one lamp to like xenon lamp to hydrogen deuterium lamp, it changes. So, what happens is basically during that region there is a bit fluctuation in the UV spectrum. Okay. So, this one is for curcumin. The intensity what we are getting out here, around the maximum peak is around 423 nanometer, so 0.023. However, this intensity is very low as it should be expected. So, what we can do, we can actually increase the amount of curcumin out here. So, we can basically double this amount or take a bit higher amount of curcumin in this case. Now, we are increasing the amount of curcumin concentration out here. Initially, we did it with 10 microliter, but right now we will take 10 times higher, that is 100 microliter. For that, we have taken this 200 microliter pipette and made the volume to 100 microliter, and I will be initially pipetting out 100 microliter from here and will be adding this curcumin. Okay. So, it is better before adding to take this solution and mix it thoroughly. Okay. You mix this thoroughly. And after that you take 100 microliter of curcumin from here and put it in this cuvette and mix it properly. Okay. The solution will turn faint yellowish out here. So, once we have mixed it, immediately transfer this one in the cuvette holder, and after that what we will do, we will directly go to auto zero. After auto zero, we will click start. So, let us increase this one to a volume value of around 0.5. Okay. So, here we can see the absorbance increases because we have increased the concentration, so absorbance will increase. The maxima comes just around 0.2, which was expected basically. There is a decrease in the absorbance value again around 350 or something like that. And after that there is again an increase with a small hump of around just over 230 region or in that case. So, let us see from here we are first deactivating the initial one and we are initially seeing that where is the hump coming. So, out here the hump is coming around given here 419. Okay. So, this peak is around 419 nanometer. Okay. And we are currently interested in this peak, or we might be interested in some other peak that we will find out later during the experiment. For the time being, what we will do, we will save this one, we will save as here as not gallic acid, or rather curcumin. And curcumin only. Okay. We are saving it out here. we are also saving it as a text file. Okay. So, this is the UV visible spectra of curcumin. Now, what we will do, we will slowly add HSA to it and see how the spectra changes. I have taken HSA solution. So, this HSA I have prepared, it is a 1 millimolar HSA, and to this from this HSA I will basically take 5 microliter of HSA. I have taken a clean micropipette once again, and I will be taking 5 microliter of HSA from it. Taken 5 microliter of HSA. Okay. Now, what I will do, I will take out this cuvette. As you can see, this cuvette is yellowish because it has curcumin in it, and I will add directly to it HSA. Okay. Why? Because this is a titration method actually. In this method, what we do, we basically go on increasing the concentration of HSA like this. We keep on adding volume of HSA to it. So, initially we have added 5 microliter of HSA and mixed it thoroughly. Please note that bubbles are not formed. Here a few bubbles have been formed, which I am actually driving out. Fine. So, once we have mixed this, I am transferring it to this chamber, and after that what I will do, I will give this auto 0, and after auto 0 I will click this start. So, let us see how it comes actually. So, the absorbance increases here, as we can see. The absorbance will again increases. In this region, and shows an almost straight peak out here. This is actually a plateau region, and it increases. Let us increase this value a bit to 1.0. Okay. So, what actually happened is that we can see that this value, the absorbance increased a bit, but this absorbance with this spectrum is not the correct spectrum. Why? Because we have to actually subtract the spectra for HSA from it. Okay. Now, we will see this similar experiment, this similar one only in a different way. In what way? Now, this is the cuvette, okay, and this is a cuvette cap. I am putting it here. This is a cuvette cap, as you can see. So, it is better sometimes to actually mix it using this cuvette cap. Fine. So, first I will actually wash this cuvette cap a bit. I have washed this previously also. Just to be safe, I am washing it currently. Was this cuvette cap, and now I am just mixing it in a different way, in which I am just inverting it. There is a risk, do not leave this cap, or otherwise entire solution will come down, and now after that transfer it in the sample holder. Close this one, give auto 0. And again start this experiment. Now, let us change this thing a bit from 0.1 to 0.5. Interestingly, what we can say is that this reading changes a bit. Okay. How it changes? The absorbance for the same solution, okay, for the same solution, the absorbance value was initially higher than that of curcumin. Now, when I mixed it in a separate way, it coming in a lower way. So, these are what? These are actually experimental anomaly which occur from time to time. How you mix it, how you prepare the solution actually determines how the experiment will carry out. Okay. So, in this case what happens, the general rule is to actually mix it properly during protein ligand interaction, take it, close it with a sample compartment or the sample cap or the cuvette cap, and then mix it and then go for observation out here. So, here we can see there is a bit decrease or almost same out here, the shift, the peak are almost same, but however, there is a slight shift in the peak intensity in the blue region, or what we call, sorry, in the red region, what we can say is the red shift. If I am not, if sure, yes, okay. So, I just confused it with the red and the pink purple line. So, the red line or the orange line actually for curcumin, and this purple line what we can see here, this line has actually shifted in the red region, that is the higher wavelength, and this one is for HSA plus curcumin. So, what has happened out here is that there is a shift in the lambda max value, okay, a bit bathochromic shift, and this region here we can see it is a bit lower than 300, and this is somewhat around 276 nanometer. This is due to the protein present here, but again what I can say is that this is not the actual spectra of the protein ligand complex. Why? I will discuss it a bit later on. Okay. Now, I have again taken this mixture. It contains now curcumin plus 5 microliter of HSA, and I will again add another 5 microliter of HSA to it and mix it thoroughly. So, the total volume of HSA out here is now 10 microliter. And similarly, the final volume has increased from 3000 to 3010 microliter. Now, I have kept this cap again once again here. This is the cuvette cap, I have kept it here, and I am just inverting it, mixing it carefully. Please note not to leave it. Okay, this is quite a bit of what I can say, a delicate process. If it slips from your hand, it might make a blunder. So, hold it thoroughly, use your thumb in this way. Okay. In the lower part, give two fingers, the index finger and the middle finger in this way, in the cap, hold it, put your hand in this way. Okay. Why? Because if it suddenly falls, the cuvette will fall in your hand. Okay. So, mix it thoroughly. And now after that, transfer this one into the sample holder, get autoflow. Autoflow done, and start with the experiment. Here the peak comes here. Okay. The absorbance actually a bit increase in the absorbance as we can see. What I will actually do is that, what is necessary to be done is that after this one, I will be subtracting the HSA absorbance. Now, why I am subtracting the HSA absorbance? Basically, what happens is that to it you are adding HSA. Okay. Initially, you have curcumin. To curcumin, you are adding HSA, and you are increasing the concentration of HSA. Now, whose absorbance are we actually bothered with? We are actually bothered with the absorbance for HSA only, or rather the absorbance for curcumin only, not HSA. Okay. So, we are bothered with the absorbance of curcumin only. Now, this spectra, as we can see here, has actually the overlap of both HSA and curcumin. Again, what I am saying, this has the tendency of overlapping of HSA as well as curcumin. Okay. So, in order to remove the contribution of HSA from here, what we need to do, we need to actually subtract the absorbance of HSA. How to subtract this one? We have to take blank absorbance after this experiment. Now what I have done out here is that I have actually taken the spectra for curcumin with HSA. It is suppose it is kept as sample 1. Okay. Now in blank actually it is sample 2, which will have the absorbance for HSA only. So your net absorbance would be A equal to S1 minus S2, or on a better note what we can say is that we have S1, which is actually curcumin plus HSA minus S2, which have only HSA. So what do you get? Curcumin plus HSA in 1 minus HSA is actually curcumin. So when we will subtract the absorbance of HSA from here, we will get the absorbance of curcumin at that particular concentration of HSA. Now, while we do the control, we can see how we can proceed with the blank subtraction. For the timing out here, we have got this green type of spectrum, as we can see here. The absorbance is a bit again higher than that of curcumin, around 420 nanometer, and this peak is again higher from the previous one because the concentration of HSA out here is much more. So I am saving this one as curcumin HSA 10, that is 10 microliter, and also in the text file. This sort of saving the spectrum file you are quite familiar with. So I am saving this one, and now I am proceeding for the next observation by adding again 5 microliter of HSA. Now I have again taken this HSA in a fresh micropipette or fresh micro tip. So please note that don't use the previous this tip, okay, while you are taking a new solution. Always use a fresh tip because the previous tip might be contaminated or it should be contaminated. So I have taken a new tip and taken the HSA solution 5 microliter, and I am adding this one. So the total volume comes to around your 15 microliter. So I am again putting this cap and mixing it. Thoroughly. Once I have mixed it, I am keeping it in this sample compartment. Getting auto zero done. Auto zero complete, and let us start. So if I can actually change this maxima from 0.5 to around 0.3, which is a bit closer out here, we can see the changes. Now this value is actually this blue line which is going out here. So more or less what we can say is that the absorbance, change in the absorbance around 423 nanometer, okay, doesn't change that much, it remains more or less same. Sometimes increase, sometimes decrease, but however this is not the final change. Once again I am saying this is surely to change 0.5 this peak, because we are increasing the concentration of HSA, and this around 278 is for the tryptophan moiety. Concentration increases. Now I am saving this one as 15. Okay. So 15 microliter. Now I will be adding to it again 5 microliter. Okay. For that I will be taking out this cuvette, taking a fresh micropipette. Okay. This is the HSA solution. I will be taking out 5 microliter of HSA from here, putting it, mixing carefully. placing this cap once again, and in up and down motion I am doing it, and finally keeping it here. After auto 0, it will give this absorbance value as 0.00, and once done, you are ready with the scanning. So, it is a bit cumbersome out here. So, all the spectra are overlapping one above the another. So, it is quite difficult for you to see. So, I am just actually removing the other spectra which are appearing, which have appeared previously actually. So, this one is for 10 and this one is for 15. So, here we can see for this 20 microliter this is coming, and this one for curcumin only, where only curcumin are present. So, again it increases a bit. Okay. So, this is for curcumin along with HSA having 20 microliter. Now, the next HSA I will be adding is 30 microliter, not 30 microliter, rather I will be adding 10 microliter, so that the total volume goes up to 30 microliter. Here I am saving this one as curcumin HSA 20 . In text file also. Okay. Now, I am taking again 10 microliter of HSA from here. Okay. 10 microliter of HSA. In this solution, I will be putting 10 microliter of HSA. Taking this cap and mixing it. So, another importance of mixing is that curcumin, as I have said, is not that much soluble in water. So, what happens if you look it carefully, you can sometimes this residues of curcumin can be found in the lower bottom side of the cuvette. Okay. So, it is better to mix it thoroughly, so that the entire curcumin present out here comes up in the solution and interacts with protein. Otherwise, we can't actually get a proper or correct result. So, we have started it. This one shows for the previous one, that is for 20 microliter. And this one goes for your 30 microliter. The next reading which we are interested in is actually 50 microliter. Rather, we are taking 20 microliter from here, so that the final volume is 50 microliter. So, one interesting thing we can see out here is that there is one crossover point where the intensity again decrease and again increase. So, there is a crossover point out here. So, this one is for our only curcumin, the red one or the orange one. So, let us do save as 30 curcumin HSA 30. Okay. Curcumin HSA 30, and now I will be adding directly 20 microliter of HSA to it. Here I am using actually 1 millimolar of HSA. For your convenience, you can also go to 2 millimolar of protein. Okay. Now I will take this out. Add HSA to it. put this cap, okay, and gently mix this one. After mixing this one, I am going to transfer it into this compartment. And after auto 0, we will give it for scan. Now, for the time being, I have completed from starting from 0 concentration and 0 volume of HSA to 50 microliter of HSA out here. Let us remove the previous ones, so it will be easier for you to visualize. So, what I will do is that now for blank or blank control, I will take a cuvette, I will fill it with phosphate buffer as we have done during baseline correction. We will take 3 ml phosphate buffer without curcumin, and then to phosphate buffer we will take 5 microliter of HSA. First we will take the absorbance, then again 5 microliter of HSA and again take the absorbance. So, basically what we are doing, we are repeating this same titration type experiment, but without curcumin. So, at each and every concentration of each and every amount of HSA, we are getting a particular absorbance graph, okay, for the protein, and what we will do, we will actually subtract this one, those graphs from these graphs, okay. So, for a particular concentration of HSA, suppose for 5, we will calculate the concentration later on from the total volume. For the timing, what we can say, suppose for 10 microliter of HSA, what we shall do is that we will take that spectra which has curcumin plus HSA 10, that is 10 microliter of HSA, and from that we will subtract HSA only 10, that is solution without curcumin only 10 microliter of HSA. So, out here we will just extend this one to 0.8, okay, and we will save it as curcumin HSA 50 and save as curcumin HSA 15 text file. So, I am now actually enabling all the files which you have currently done till now, 1.0. Okay. So, this one is for only curcumin, okay. The second one is for curcumin, and this one HSA 5 microliter with curcumin. The third one is for 10 microliter, the fourth one out here, the fifth one for 20 microliter and 30 microliter, the sixth, and finally 50 microliter. So, what we can see is that there might be a bit increase out here in the change, there is a bit increase in this region, but however, there is a strong change out here. So, no need to bother regarding this because these are all the protein peaks. Now, coming to this region below 250, here this is 200, this is 300, this comes around 276, and coming to region 200 to 220, if we just extend it to around 2.0, more than that, 0.0. So, these are all actually the peptide, this comes for the peptide absorbance 4.0. So, it is actually a very high absorbance value. So, here you can see for pure curcumin, here pure curcumin, this one, this which one is appearing and disappearing right now, this one is pure curcumin. So, it does not have any such peaks, okay. These peaks, these are for peptide, and once you have started adding HSA, all these peaks are coming. So, we have to remove all the unnecessary peak to get the absorbance of curcumin, okay, absorbance of curcumin in presence of protein only. So, I am transferring it to 0.5, and now we will proceed for our control spectra. Now, we are doing the experiment, the absorbance for blank subtraction method. We have taken this same cuvette which I have kept in the sample compartment, we have washed it thoroughly with double distilled water. Please make sure that curcumin is not present out in the cuvette. You can actually check it with your eyes only, because if curcumin is present, it will appear yellowish. If you still feel that you can't actually remove it with double distilled water, this yellowish tinge, add just ethanol to it, and you can add at that time your washing ethanol, that is the local ethanol, not the high graded ethanol, and you can wash your cuvette properly and you can proceed for this observation. And after you have washed with ethanol, please make sure to dry this cuvette and wash again with double distilled water. For the time being out here, the entire solution, I hope it is completely removed, because I can't actually see any sort of yellowish tinge out here. I washed with double distilled water, and now I will pour here 3 ml of phosphate buffer